Welcome back, my dear students. Today we will learn about gluconeogenesis. Gluconeogenesis or neoglucogenesis means new creation or formation of glucose other than carbohydrate sources. Basically, it's the process that makes glucose or glycogen from non-carbohydrate sources when carbohydrate in our body is depleted or low, such as during starvation or intense exercise. In simple terms, we can say gluconeogenesis is like the reversing glycolysis, which is the breakdown of glucose for energy, and glycolysis converts glucose into pyruvate, which then enters into the citric acid cycle. Gluconeogenesis starts with pyruvate and converts various substrates into glucose. However, three key steps in gluconeogenesis are different from glycolysis, and they require specific enzymes, allowing the process to occur even when glucose are very low in our body. So when fasting in the body, it maintains glucose using gluconeogenesis. It's a metabolic pathway to make glucose from amino acids, lactate and glycerol. It is primarily occurs in liver cells, but it is also found in renal cortex of the kidney and in the epithelial cells of the intestine. So gluconeogenesis, gluco means glucose, neo means new, and genesis means production. So we know the synthesis of glucose from non-carbohydrate sources. Basically what happens, carbohydrates after in digestion through the enzymes in our digestive system, it break down and it forms glucose. But other than carbohydrate, fat, amino acids, pyruvate can also form glucose. So the formation of glucose from the sources other than carbohydrates are called as gluconeogenesis. Sites of gluconeogenesis: the liver and the renal cortex of the kidneys are the main sites for gluconeogenesis, as they contain the necessary enzymes including glucose 6 phosphatase to release glucose into the bloodstream. The liver mainly uses lactate, alanine and glycerol for this process. Gluconeogenesis can occur to a small extent in the small intestine during fasting. So in our body, in liver, 50 to 60% gluconeogenesis occur. In kidney, there are nearly 40% gluconeogenesis occur, but intestinal epithelial cells participate in gluconeogenesis, but that is very minor. So the cellular site for gluconeogenesis is the cytosol and mitochondria. So what is the concept? When we intake our food, after digestion, this food produces glucose, and the blood glucose level in our body usually found between 100 to 200 milligram per deciliter. But when glucose is excess in our body, it will be deposited as glycogen in our liver. But when glucose level fall, then glycogen is depleted and it produces glucose within the blood. But fat, amino acids, pyruvate can also produce glucose during fasting. In our nervous system, the brain cells and the RBCs are the tissues where readily uses the glucose for their nutrition. Concentration: when glucose, basically 6 carbon compound, and after 10 reactions, among which 3 are irreversible reactions, it will produce 3 carbon pyruvate, and then pyruvate, after pyruvate again can produce glucose due to presence of specific enzymes, and it can reverse these reactions, which are upregulated during fasting. Lactate will produce pyruvate, and this pyruvate will produce the glucose, and propionate will form the Krebs cycle, and from the Krebs cycles it will again produce the pyruvate, then pyruvate will again produce the glucose. So what happens during gluconeogenesis? The pyruvate, which is produced after glycolysis, it comes into the mitochondria, and in presence and within mitochondria, with the help of ATP, it releases one molecule of carbon dioxide, and it will form the oxaloacetate. The oxaloacetate then again moves from mitochondria into cytosol, and with the help of the enzyme phosphoenol pyruvate carboxykinase, it will form the phosphoenol pyruvate. During this process, one GTP is utilized to form one GDP, and one carbon dioxide molecule is again released. and from fructose 1,6 bisphosphate, with the help of fructose 1,6 bisphosphatase, fructose 6 phosphate will produce, and with the help of glucose 6 phosphatase, from glucose 6 phosphate glucose is produced. So these are the four reactions of gluconeogenesis which occurs as the three reactions of glycolysis is irreversible in nature. So now we have to know that is gluconeogenic amino acids undergo transamination, which causes change in the carbon skeleton and directly gets converted to pyruvate. Some glucogenic amino acids from oxaloacetic acid or other intermediates of citric acid cycle, they can participate in gluconeogenesis, while alanine is preferred in liver, glutamine is preferred in kidney. Lactate, muscular activities and anaerobic glycolysis in red blood cells produce a large amount of lactate. This lactate is taken up by the liver and gets converted to pyruvate by the enzyme lactate dehydrogenase. Pyruvate then undergoes converted to lactose, converted to glucose by hepatic gluconeogenesis, which is then sent back to muscles for reuse. This is also known as Cori cycle. Glycerol is formed by breakdown of triacylglycerol in the fatty tissue. It is then carried to the liver where it gets converted to pyruvate and enters gluconeogenesis. So now we need to know regulation of gluconeogenesis. The body carefully regulates gluconeogenesis or glucose production and glycolysis, that is glucose breakdown based on energy needs. When there is excess glucose, the body uses it for energy or stores it as glycogen and fat. When glucose levels are low, the body produces glucose through gluconeogenesis using non-carbohydrate sources like amino acids, lactate and glycerol. Since gluconeogenesis is almost the reverse of glycolysis, both the processes are regulated in opposite directions when one is active and the other one is inhibited. This balance is crucial for maintaining stable blood glucose levels and is influenced by substrate availability and hormonal signals. Role of glucagon in gluconeogenesis. Glucagon is secreted by the alpha cells of the pancreas. It plays a key role during fasting or low blood sugar. It stimulates gluconeogenesis through the following mechanisms. Inhibition of fructose 2,6 bisphosphate formation. When glucose is high, fructose 2,6 bisphosphate inhibits fructose 1,6 bisphosphatase, showing gluconeogenesis. During fasting, glucagon decreases fructose 2,6 bisphosphate, removing this inhibition, promoting gluconeogenesis. Thus, glucagon acts opposite to insulin. Inactivation of pyruvate kinase. Glucagon inactivates pyruvate kinase by phosphorylation. This step slows glycolysis and directs the metabolic flow toward gluconeogenesis. Increased enzyme production. Glucagon enhances the production of phosphoenol pyruvate carboxykinase or PEPCK, a key enzyme in gluconeogenesis. This increases the pathway capacity to produce glucose. Role of glucocorticoids in gluconeogenesis. We know glucocorticoids or cortisol are stress hormone produced mainly by adrenal cortex, not the kidneys. During stress, fasting or intense exercise, they help maintain blood glucose by stimulating gluconeogenesis in the liver and kidneys, increasing the breakdown of proteins into gluconeogenic amino acids which serve as raw materials for glucose synthesis. This is why cortisol is often called a glucose sparing hormone, as it ensures a steady glucose supply during energy shortage. Now we need to know role of insulin hormone in gluconeogenesis. Insulin basically secreted from the beta cells of the pancreas acts as the main anti-diabetic hormone that lowers blood glucose. Here, how insulin regulates glucose metabolism. When blood glucose rises, beta cells release insulin. This happens because increased glucose causes higher ATP production, which affects potassium and calcium channels, leading to insulin secretion. Insulin inhibits gluconeogenesis, thereby preventing excess glucose formation. It promotes glucose uptake by liver, muscle and fat cells for energy use or storage. Hormonal interactions. Epinephrine or adrenaline inhibits insulin release during stress. Glucagon stimulates insulin release after meals to balance blood glucose. Overall, insulin reduces gluconeogenesis in both the liver and the kidneys. So, my dear students, today we explored the fascinating process of gluconeogenesis. That's the body's remarkable way of making glucose from non-carbohydrate sources like amino acids, lactate and glycerol. We learned how this process works mainly in the liver and kidney renal cortex, ensuring a contrast supply of glucose during fasting, starvation, or intense exercise. We also understood how hormones like glucagon, cortisol, and insulin finally regulate the process to maintain blood sugar balance and keep our body's energy system running smoothly. If you enjoyed learning about how the body creates glucose even in the absence of food, don't forget to like, share, and subscribe for more such interesting and easy to understand biology lessons. See you in the next class, and as always, stay curious, keep learning, and keep exploring the wonders of life. Thank you.